an important PYQ for us, and the PYQ relates to biochemical test. We shall have questions which is important and has appeared in earlier competitive test examinations, and the biochemical test which we shall perform, and we shall have an idea about the biochemical test relating to this PYQ is about Zalbinoff test and also xanthoprotic test. Now let us see the question. The question is here. Zalbinoff test and xanthoprotic test are used for identification of course, blank spaces are there and two blank spaces respectively. We are required to fill up these blank spaces as given in the options. 4 options are there and we have to find out the correct option which has to be filled in blank space first and the other blank space second. So here, of course, it is a knowledge-based question and we get the answers very correctly that in this case, the option 1 is correct. And therefore, the correct answer is option 1, ketoses and proteins. The other options are not applicable over here. Well, we got the answer this way. It is a knowledge-based question and our studies do not complete here. Simply answering the question will not be effective to us so far as our learning is concerned. And therefore, we need to learn what is Selivinoff test and what is xanthoprotic test. The important points of Selivinoff's test and xanthoprotic test we need to know. Now, therefore, let us move to learn what is Selivinoff's test and what is xanthoprotic test. Now here it is. According to Selvinov's test, we use a reagent which consists of resorcinol and concentrated hydrochloric acid. When aldoses and ketoses are to be distinguished, what we do that aldoses containing a keto group, that is CO group, when a solution of aldoses is mixed to this reagent, we get a deep red color like this. And this color represents the presence of ketoses. And it is a very quick test. So what happens, this test relies on the principle that when heated, ketoses are more rapidly dehydrated than aldoses. So certain points are provided here, and this is a foolproof test and quick or sharp test. To credit goes to Theodore Selivanov, the chemist who devised this test, and the quick appearance of the red color like this. This is known as cherry red color. Cherry red color like this represents the presence of ketoses. Now, aldoses also undergo this reaction, but the reaction is not fast and we get a faint pink color or light pink color this way. Now, what is the reaction taking place is given over here. What happens? The dehydrated ketose, one acid hydrolysis of ketose yields simpler sugars followed by furfural. The reaction is here. When you dehydrate a ketose in presence of acid, we get a furfural form. And then when this is treated with resorcinol in presence of an acid, we get a compound which has a deep red color like this. Generally, as given here, 6-MHCl is used for this test. Ketoses are dehydrated faster and give a strong color like this. Whereas aldoses means those sugars which contain aldehydes. Ketoses, these are those sugars which contain ketonic group, CO group. Well, all this is known to us, and these are certain points which we need to go through so far as Savinow's reagent is concerned. Now, next we move to what is known as xanthoprotic test. Now, the xanthoprotic test is very simple test, and it is a test for presence of proteins. You must have worked in the laboratory using nitric acid, concentrated nitric acid, and you must have observed that when we work with concentrated nitric acid and accidentally or unknowingly nitric acid gets in contact with our skin turns brownish area or yellowish. This is simply because the skin contains protein, and protein, when it comes with uh, our strong acids like nitric acid, it turns yellow color or orange color. The intensity of the color is directly proportional to the amount of the protein present. It is only a representative image. और आपने देखा भी होगा कि जब आप लेबोरेटरी में या प्रयोगशाला में काम करते हैं और भूल से थोड़ा सा नाइट्रिक एसिड, कंसंट्रेटेड नाइट्रिक एसिड आपकी उंगलियों में लग जाता है या स्किन से कांटेक्ट में आ जाता है, तो आपकी स्किन में कुछ लाल या भूरा सा रंग बन जाता है. ये क्यों होता है? इट इज सिंपली बिकॉज our skin is nothing but built up of proteins, and this protein reacts with nitric acid and it gives a yellow color. So it is a common observation of the xanthoprotein test. Okay, thank you very much.